Good evening. There's a lot of you. Welcome to Café Scientifique, put on by the Faculty of Science at the University of Victoria. Before we get started, I would just like to acknowledge and respect the Lekwungen people on whose traditional territory the university stands, the Esquimalt, Songhees, and Wasanic people whose historic relationship with the lands continued to this day. This is also where we're able, the traditional lands is also where we're located now and able to talk about science with the community. And thank you very much for coming tonight. You're in for a treat. Our speaker tonight is Dr. Julian Lum. Julian's foray into science started when he was young. Apparently he was very interested and almost obsessed with ornamental fish, actually. And as a young lad, he had 11 to 13 aquariums in his house filled with ornamental fish. And as any pet owner, some pass away, others live longer, and you know, understanding why that was kind of what sparked his interest in science. It also must have sparked his interest in being in the water, because he's also an exceptional swimmer and held the 50-meter and 100-meter record in front crawl for a couple years. So, yeah, so he's multi-talented. Now, fast forward to today, he's a distinguished science in cancer immunology. He is located at BC Cancer. He's a professor at the University of Victoria in the Department of Biochemistry and Microbiology. And he is also director of the Metabolomics Consortium of British Columbia. And he truly is a star in cancer immunotherapy. So please join me in welcoming Dr. Julian Lum to tell us a little bit about how metabolism, how it can help or hurt cancer therapy.
Okay, thank you so much, Jen, for that really kind introduction. Thank you all for coming tonight. Happy holidays, in case I forget to wish you happy holidays at the end. So tonight, I want this to be a participatory event. So I brought a little brown bag. And so be careful of the person who has the little brown bag. And I'm going to pass around a couple of props. And I'd just like you to have a look at them. And while you're looking at them, think about how much one of these would cost, okay? Because I'm going to get to, towards the end of my talk, a little bit more on like, how do we do research? Why does it cost so much? And how do we get our work funded, okay? So I'll just start at the top here and I'll just pass it around. So have a look and I'll tell you what these items are later on this evening, okay? Okay, so I'm going to start off with a question that I ask my students whenever I give the lecture on cancer biology. So the question is, how many cells does an average person have in their body? So if you know the answer, you can raise your hand or you can shout it out. What do you think is the magic number? Billions, okay, trillions. Any other guesses? 2 trillion, okay. So raise your hand if you think it's in the billions. Okay. And raise your hand if you think it's in the trillions. And raise your hand if you don't really care. Okay. So on average, an individual has about 40 to 50 trillion cells. Okay? So that's the magnitude of the problem that we have to face when it comes to cancer. And that is that any one of those given cells can become cancerous at any time in your life. And so the question becomes, how does the body keep all these cells in check so that you don't develop and go on to develop cancer? Okay? And so that becomes the magnitude and the question that we're asking. And so tonight's talk, I want to have basically two topics that I want to touch on. And the first topic is, how can we use our own immune system, our own body's defenses, to be able to treat the disease of cancer? And then the other part I'm going to touch on and talk to you about is, how do we ensure that our immune system is primed, is ready to fight that battle at any given moment in time? And so the way that I think about this is, we need an approach where we can be able to change the way our bodies see cancer, and that those cancers can't hide out in places. And so the army, if you will, needs to be able to go out and seek out all the places that cancers can hide in, all those 50 trillion cells that we have in our body. Okay? So I'm going to start on the topic of, systemically, how can we think of ways that we can do that? So raise your hand if you've ever done a cancer experiment. Okay, we got some people in the audience here. So I surmise that every person in this room has done a cancer experiment. Okay, and the cancer experiment is, when you were a college student and you left the bread out on the countertop for many, many days. And so you know that what happens is that some spores of mold latch onto the cancer, or latch onto the bread, right? And what those spores will do is they will grow and divide, and continue to grow and divide until there's no more nutrients for those spores to grow. And then what happens? Those spores jump, they move, they metastasize to another piece of bread, or another part of the bread where there's nutrients, where there's things that they can eat to fuel the growth and the division of those spores, until there's your entire bread is completely covered, right? Okay, so what are the nutrients that our cells need in order to grow and divide? Protein, good. And in scientific terms, we classify the proteins as these metabolites, which we call amino acids. What else do we need? Sugar, beautiful. And in scientific terms, we don't call it sugar, we call it glucose. What else do we need? Yes, we need some ATP that we derive from these proteins and these sugars that we eat. Okay. So one of the things that we have uncovered during our scientific endeavors is that glucose is the prime source of fuel for cancer cells. Okay? And we know this because there's anecdotal evidence and direct evidence that there's a correlation between the incidence of type 2 diabetes and cancer, almost a perfect correlation, right? And so you ask the question, well, how are these two interlinked, okay? All right, so here's the next thing that we got to do together. So I want you to pull out your cell phones, okay? So everybody pull out their cell phones. go to a browser, okay? And in your browser, you're going to type Windsor salt ingredients. So Windsor salt, right, in the little blue box with the checkered. I want you to type that into your browser and then click on the image and somebody tell me, what are the ingredients in Windsor table salt? And sugar. So why is there sugar in salt? Okay. All right. And so now I want you to take your cell phone, spin it around, and do a selfie with me. Okay. All right. So this is a problem because, you know, the food industry in particular has supplemented many of our foods with sugar in places that we don't even know about. And what does the sugar do? What's the first thing that happens to your body when you have a meal that has sugar in it or carbohydrates that can be broken down into sugar? What's one of the first things that your body does? Insulin, right? Banting and Best discovery, Canadian discovery. Your body secretes insulin. And what does the insulin do? Exactly. You have sugar in your bloodstream, so you get a spike in your bloodstream level sugar, which you can detect with one of these glucometers, right? Insulin is secreted, and the sugar, the glucose, now goes into the cell. And now the cell has a source of fuel to begin to divide and begin to grow. So every time you have a meal and you have a sugar spike, insulin is the signal that tells your cell, we got lots of food, let's start growing, okay? So, you know, we, throughout history, right, have changed in terms of our level of physical activity, right? Hunter and gatherers, where there were many periods where food was not available, readily available to us. We had to go out and hunt for it or gather it, okay? And over time, we reached a point where we were, hit this agricultural revolution, right? We began to use tools, we began to have cattle and et cetera. And so food became much more readily available, right? And then finally, I think the thing that really nailed the coffin for being able to, be in a position where you could just get up from your couch and get food is refrigeration, right? And it was at that point that we began to see this incidence of many metabolic diseases such as type 2 diabetes, obesity, and then the coincident rise in cancer as a result of those, right? So one of the things that we've been trying to understand then is if there's this pervasive amount of glucose that's in our body system and it's fueling our cells to grow, then how does that cause and trigger cancer cells, right? Normal cells do this, normal replenishment of cells that are dying, et cetera. So the question became, well, what is the basis of cancer then? If we're seeing this concordance, this relationship between, diabetes and cancer incidence. So what is the cause of cancer? Just thinking about what you've heard, what you've read, what you know.
Something goes awry in the immune
Okay, good answer. Good answer. So something goes awry in the immune system, can't detect the cancer cell. What other things cause cancer? Mutations. Excellent. Okay. Anything else? Runaway growth. Okay, good. So I want to get back to this point on mutations. So how do mutations come about in a cell? Does anybody know? Yes, that's a component of that, for sure. Yeah. DNA mistakes. That's kind of the definition of the mutation in our genomic information. So every time a cell divides, it has to copy its entire genetic code to give it to its daughter cell, right? And so every time you try and make a copy, nature's not perfect, so it can make mistakes and mutation, right? So that's one way that it happens. Another way that it happens is that there's these little organs inside the cell, which we call the mitochondria. I don't know if you've heard of that. If you remember your biology 101, they're the powerhouse of the cell. They make the ATP. One of the byproducts of an active mitochondria, right, that's making ATP, the energy that's required for cells to divide, one of the byproducts of that is something called reactive oxygen species. And these are toxic molecules that damage DNA and that cause direct mutations, right? So you can have errors, but you can also have mutations that come about through direct damage through these reactive oxygen species. So every time your cell divides, it's going to be prone to these mutations, right? But it needs the food, it needs the fuel in order to initiate that event of division. Okay. So I'm going to shift gears a little bit and talk about, well, how is this related to the point that was made here about your immune system? Okay. And, how is the immune system now linked to this cancer growth that's fueled by, these sugars, et cetera, in your body? So one of the things that we've learned in the lab is that excess sugar can actually cause your immune system to become suppressed, okay? Excess fat intake, excess protein intake can have similar effects. And so there's been a lot of work now looking at how can we modify your diet to reduce these suppressive effects on your immune system, okay? And I'll tell you one story that we've been working on that we're very excited about, which is a study that we've conducted in the last year where we enrolled patients that have a leukemia, and we put them on an intermittent fasting diet, okay? And there are various forms of intermittent fasting, and the one that we chose was called 16-8. Okay, some of you might have heard this. So there's a 16-hour period where there's no food consumption. Liquids are fine, clear liquids, that's okay. And then there's an 8-hour window where we do not restrict what or how much the patient eats. Okay, and then they go back onto the 16 window where they don't eat. Okay, sometimes we call this time-restricted eating. That's another term that you might have heard. And so we did this study with two goals in mind. The first goal in mind was, what happens to their immune system? What kind of changes can we see? Is the immune system more activated or less activated? Okay, so that was goal number one. The second goal was we wanted to understand if there were changes in the gut microbiome. Okay, and the gut microbiome is essentially the healthy bacteria that live inside your gut. And there's been a lot of work now linking the health of your gut microbiome to diseases, including neurodegenerative diseases. There's been a lot of work on cancer, and we're now just scratching the surface to ask, is the gut microbiome also altering the way that our immune system behaves? Okay, so what did we find? Okay, so the first thing we found was there are certain types of white blood cells in your body, which we call T cells, okay? And these T cells, their job is to seek out foreign pathogens, viruses, bacteria, parasites, etc., okay? And we measure them directly out of the blood in the patients who were on this intermittent fasting regimen. And what we found was that not only did the number of T cells increase, we also found that those T cells could actually kill cancer cells better in a Petri dish. Okay? And so the question now is how and why, and that's something that we're actively working on. We want to know the real molecular basis for why this shift in just the time that people eat can cause such a profound change in their immune system and their immune cells. Okay? Okay, so the other part of the equation is what happened to the gut microbiome. And so the way that we did this is we analyzed the metabolites. So these are little molecules like amino acids, etc., but they're not produced normally by our human cells. They're produced actually by the bacteria that reside in your gut. And so we looked at that. We analyzed what is being produced by the microbes and the gut microbiome. And we found two very interesting things. And the first one is we found a metabolite. I won't give you the name, it's a big long name, that we think actively can promote the immune system. So we think maybe that this is the key. The key is that the bacteria in your gut in these patients were producing this metabolite that was directly acting on the T cells in that patient. Okay, what was striking is that 65% of the patients that were on the study, and I want to caution because this was a very small study. We had 18 patients that were enrolled on the study, okay? So these studies need to be replicated in larger patient groups. But in these 18 patients, 65% of those patients, we saw that their leukemia cell counts, so the cancer cells in their blood, either went down, was flat, or didn't change, right? And so, again, a very small study that we need to replicate, but it gives us some hope that maybe we can alter the immune system in a way that protects them, the patients, from the malignant cells or the cancer cells from growing and dividing. Okay. All right, so another very interesting and exciting area is, I'm sure you've all heard of ketogenic diets, right? These are diets that are predominantly very high fat, low carb, and certainly we've seen a lot of very promising changes in people in terms of body composition, right? And so we've been trying to understand now, is there a link between the ketogenic diet and a person's immune system? Is it promoting it? Is it helping it? What's it doing? Okay. And so we've been working with colleagues in the US, in Michigan, where we did a small study and we put a group of patients on a ketogenic diet for 3 months, and then another group of individuals who had just their normal diets, okay? And again, the key thing that we measured was their immune cell function, their T cell function, and their microbiome. And to make a long story short, is that we identified this very interesting metabolite that increases. And this metabolite is called beta-hydroxybutyrate. It's something that is produced naturally, but also by the gut microbiome. And this metabolite on its own could enhance the ability of patients to fight infections, okay? Because we track that over time. Again, very small study. There were 25 patients on this study. But what we did was we took some of those patients' T cells and we actually, in a Petri dish, we added this beta-hydroxybutyrate. And what we found was quite striking. Those T cells that were in the Petri dish grew and expanded exponentially. Okay. All right. So this is kind of the state of where many of us in the field are really driving towards is how are these diets and how are the changes in our diets affecting our immune system, and how can we learn from that and perhaps use diet now as a tool to alter the course of a patient that might have cancer. And we're still a long ways there. We still have lots of work to do, but that's a basic premise of the work that we've been doing in the laboratory. Okay, so the second topic that I want to talk about tonight is how can we actually reprogram these immune cells, these T cells, given the knowledge that we've been learning about these metabolites that are promoting their function. And so some of you may have heard of some very exciting technology called CRISPR engineering. Okay, this was a Nobel Prize that was won 3 or 4 years ago by 2 scientists. And the idea of CRISPR engineering is that you can go into the genetic code and just like little scissors, you can snip out little pieces and then you can replace those little pieces of DNA that you've removed with a new piece of DNA. A piece of DNA that you can program that cell now to do something that it couldn't do before, or program that cell to do something better than it could do before you made that engineering manipulation. Okay, so cancer cells have these molecular beacons, okay, that are decorated on the surface of the cell. And it tells the immune system, which has another molecular marker, to latch onto that beacon and then kill that cancer cell. Okay, so there's 2 beacons. The one that's found on the cancer cell and the one that's found on the T cell. Okay, and as aptly pointed out by a person in the audience tonight, sometimes the cancer cells can hide and they will remove that beacon so that it's invisible to the immune system. But we have tools like CRISPR now where we can engineer the immune cell to recognize other beacons that they don't naturally recognize, okay? And we call this beacon a CAR, C-A-R, and that's short for chimeric antigen receptor, okay? So now we can take a patient who walks into our clinic here in Victoria, we get a blood sample of their T cell, and then we genetically modify that T cell with a CAR that's going to recognize the cancer cell, okay? And then we grow these cells that we have genetically modified into billions and billions of these cells. And they come in basically like an IV bag. And we take that IV bag and we infuse those CARs, which we've genetically modified so that they recognize the cancer, back into that same individual. Okay. And the hope is that this army of T cells, these CAR cells, now can go and find the cancer and kill it. Okay. So one of the studies that we've done here in Victoria is for blood cell cancers, okay, leukemias. And you might have heard in the news, the CAR that we've used is something called CD19. That's the molecular beacon that these CARs will recognize on the cancer, CD19. And it's very exciting because we are here in little old Victoria. We're the only place in Canada that is currently able to make these CD19 CAR T cells and give them to patients. And the response in the patient has been outstanding, like exceedingly. More than 85% of leukemia patients who are eligible for this CAR therapy respond to the treatment. And out of those 85%, 50% remain cancer-free up to 3 years. And that's how much data that we've collected so far. 3 to 4 years so far remain cancer-free. So it's very exciting technology. It's very exciting work that we're doing right here in Victoria. Now, how do we make these CAR T cells better is the question, right? And one of the things that we learned is that cancer is a very unique beast because it evolves over time. It's almost a Darwinism kind of an approach, right? You give it some chemotherapy, some of the cells die, some of them don't because they're resistant, they might mutate, and it's a sort of a survival of the fittest, right? And what we've learned is that those surviving cancer cells begin to secrete, they excrete these metabolites that I've been telling you about. And these metabolites aren't good because it's protecting the cancer cells from the immune system. They're doing the exact opposite of what I described to you in the studies where we looked at the gut microbiome. These metabolites are shutting off the immune system, okay? And they're doing that permanently. So, how can we use these engineering technologies now to make an immune cell that is ignorant to those metabolites that are shutting off the immune system? And that's what we're attempting to do right now, is that we're trying to identify all of these metabolites and actually make the immune systems completely ignore it so that they can still function when they go back into the patient and go back into the patient's body and find those cancer cells, okay? So I think there's a really interesting opportunity now because we have essentially merging of two very important fields in cancer. And that's the field of tumor immunology and then this field of systemic nutrition or metabolism. And what my laboratory is trying to do is bring these two together so that we can leverage all of the information that we've learned to make your immune system or your T cells better at recognizing the cancer cells. So fast forward, 5, 10 years down the road, where do we see this all going? Like, how do we put all these pieces together? And one of the things that we've been doing now is combining these two things, right? So how can we use diet and nutrition, right, to make your immune system primed, ready to go, and at the same time, when we're generating these engineered CAR T cells that we're giving back to the patient, how can we have the patient in a state where those cells are going to be supercharged and ready to go and attack the cancer? And I think that's kind of the next frontier that we're thinking about in terms of where are we taking these two strategies, and how are we going to merge them together to find new ways to treat cancer. How am I doing for time here? Yeah? 7:35. Okay, great. Okay, so I'm going to pause now on sort of the more scientific part of my talk, because I want to give ample time for questions. And so those little props that I passed around, does somebody have the one with the blue top? Looks like a little cylinder. Ah, beautiful. Okay. So I often get, oh, there's one in the back there too. So I often get the question, you know, how does this research all come together? Like, you know, how do you fund your research? How do we make these advances? And so I thought what I would maybe impress upon you is a little bit about like what I do on a day-to-day basis in terms of, you know, finding ways that we can move the research forward as quickly as possible. So my laboratory has about 10 people. Most of them are students from UVic, graduate students, undergraduate students, and I have a couple of staff members. So just a wild guess, because I know this is a question that you've probably never been asked, but how much do you think one of those blue top cylinders cost? Oh, 4 cents. Wow. I wish it was 4 cents. $4.50. So, you know, before all this COVID stuff, it cost about 45 cents. But now, you know, with inflation and et cetera, it's about $1. Okay, so you're saying that's not very expensive. Okay, somebody who's got their phone out, we gotta do this again. Open up your calculator, because you can help me do some math. Okay. Who's got their phone up? Raise your hand. All right, right up at the front. So $5. I have 10 people working in the lab, so times 10. Excellent. On average, one experiment will require about 20 of those. Okay, so times 20 per day. And my graduate students, you know, I know they work really, really hard, but they do like their weekends off. So let's say they work 5 days a week. So times 5. And I know they like to have their holidays. So let's just call it 45 weeks in a year. Yeah, 5 days a week, 45 weeks. Okay, so in a year, 10 people in my lab, just that one little cylinder. How much are we at now? Come again? $225,000. So that's just one little tube. We might be a zero off there. Might be 22,000. But it's still a lot. But that's just one little cylinder, okay? And I passed around a few other things there. There's these long pipette tips, right? And then inside one of those blue top containers is smaller tubes. We call those Eppendorf tubes. You're welcome to take them out. Those are a little bit cheaper. Those are probably about $0.25 each. And so this is a very, very expensive endeavor. And often people ask, well, how can I help? I would surmise a different way. I mean, donations are always welcome. But, you know, I think that we need to inform and educate and speak to our governments about funding fundamental science and having them invest in that. And that is the most important thing that each and every one of you can do, is to support science by going to vote for the people that will, invest in this very important research. And not just in cancer research, I should say, and all research across the board. Okay, so thank you very much for your attention. I'm happy to entertain questions. Yes, great.
I'm going to moderate here. So if you say your question, and then Dr. Lum will repeat the question for the people online. Go ahead.
Do you not reuse those glass vials?
Do we not reuse them? There was a time when I was a graduate student where they were all made of glass, or many of those things were made of glass, and they were washed and reused. There was a period then of time where we transitioned to what we call one-time use plastics. Yeah, and so the short answer is there are some things that we do reuse, but by and large, they're one-time use.
Here.
When you put the T cells back in, do the T cells that are already there recognize there's something different and fight them?
Great question. So when you put the T cells back in, the T cells that are already there recognize something in the cancer or in the T cells that are coming in, right, that you're infusing. So one thing I didn't get into the details about is that before we give those genetically modified T cells that we've produced in the laboratory and manufactured in the laboratory back into the patient, we actually do a manipulation in the patient, which we call lymphodepletion. And that lymphodepletion wipes out all of the T cells that they have, so that the incoming T cells that we're infusing have space and room to grow and divide. So that's a great question, yeah.
Back at the bar.
Yes, could you repeat how intermittent fasting, what was your finding on from that? How specifically did it affect the immune?
Yeah, so what was the finding related to changes in the immune system on patients that were on the intermittent fasting study? So we found two things. And the first thing that we found is that the number of T cells circulating increased. And we also found that if we tested their T cells for their function in a petri dish in the lab, their function also increased. And they were able to recognize cancer cells in a petri dish much better than those patients who were not on the intermittent fasting.
So that's a net increase in immunity?
We believe that it's a net increase and a net benefit. And I should say that we're one of the first groups in the world to begin to explore this, but there have been other studies not looking specifically in a controlled way. So there was a study in the US looking at people who participate and practice in the Ramadan fasting. And they did not specifically look at immune cell function. They looked at other biochemical markers in the body, like changes in their cholesterol, changes in their HDL, etc. Yeah, so definitely benefits that we're seeing. But again, I do caution that this is something that we need to repeat and show in larger studies. But it's very promising.
Mayor?
Oh, yes. Yeah. I believe Himalayan salt, sea salt, kosher salt, just not Windsor salt. Sorry, Windsor.
Do you have a theory as to why the intermittent fasting created this? Is it the decrease in sugar? Is it having a longer period without any foods? I mean, there's a whole bunch of things that could be the causation, which I'm sure we can correlate.
So, do we have a theory of why this intermittent fasting caused these changes? It's a great question. And, I think our thought on this is that the gut microbiome, by the way, does anybody know how many bacteria that you have in your body? This is another. Yeah. 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 There are more bacteria in your gut than there are cells. We'll leave it at that. Okay, so, this is a very fascinating area. We don't really know very much about it. And it's very dynamic because when you eat, we do know that from a cultural standpoint, people who eat different foods have different compositions in their gut microbiome. Okay? If you were to grow a tomato in Ontario and grow a tomato in British Columbia, because the soil has a microbiome, right? And you're consuming the produce from the soil, you're also consuming the microbiome. Well, the microbiome in Ontario in their tomatoes is different than the microbiome in the tomatoes in British Columbia. So, to answer your question, we don't know. But we think and we hope that the microbiome is going to give us some clues as to how these different diets are altering the composition of the gut microbiome to enable these benefits that we see in the immune system.
Yep.
The question is twofold. One is kind of nutritional and one's more engineering with CRISPR. But it's referring to the metabolite, was it beta hydroxybutyrate?
Mm-hmm.
Is there a particular type of yogurt or something like that that promotes that particular bacteria that would produce that metabolite?
Great question, because there's certainly in the nutraceutical, so let me paraphrase the question. Is there a particular food in yogurt or some other that might contain this beta hydroxybutyrate that promotes immune cell function? So the nutraceutical industry, right, through probiotics, are trying to do this, are trying to replicate some of these things, but without knowing what exactly they should be putting in the probiotic, right? And so we're kind of taking a first principles approach and saying, well, what is changing, first of all, in the person, in their microbiome, and then figuring out how those changes are actually either improving, or there may be metabolites that we haven't uncovered yet that might actually be suppressing the immune system, right? So the answer to your question is, I don't know if there are any specific food groups or foods or supplements that contain beta hydroxybutyrate, but I'm sure we can ask Chat if there's a supplement that's out there that has beta hydroxybutyrate. But a ketogenic diet, one of the main products of a ketogenic diet is the body producing beta hydroxybutyrate.
Second part, much similar to the COVID vaccine, which would have to produce an mRNA avenue or something like that to put yourself in where I'm imagining using CRISPR substitute to code that. Is it possible that they could use a similar type of short-lived vaccine that could carry the genetic code for that metabolite?
Oh, I think you should come and join the lab. That's a wonderful idea. So the question is, can we use mRNA technology in some way to create cells that would make a metabolite that would help the immune system? And so the short answer to that is yes, but the long answer is, how do we functionalize that? How do we actually do that in the laboratory? I think that's a great avenue of pursuit. What I can say is that mRNAs are short-lived, and so when the cell takes it up and starts to generate the metabolite for that, that's encoded by that mRNA, there's a finite period of time that happens because the mRNA eventually is used or degraded. So there needs to be perhaps some sustained way to keep that going. But maybe you don't. Maybe you only need to have it for a short period of time when the person, really needs that immune boost.
Well, my thought process was when you created those special T cells for leukemia, that you could give that vaccine at the same time to boost the activated T cells.
I think I want you in my lab.
So that's exactly for the CAR T cell approach, exactly what groups are trying to do now, is to give those genetically modified CAR T cells a boost after they get into the patient's body. So one thing I want to say is that this CAR T cell approach has really been groundbreaking and changed cancer treatments, particularly in what we call the hematological cancers. All the cancers are like in the white blood cells, basically, right? Leukemias, lymphomas. Where we haven't made similar inroads is the other 85% of the cancers, which we call solid tumors or epithelial tumors, like the breast cancers, the colorectal cancers, the prostate cancers, the lungs, etc. So we don't quite understand why. Maybe it's because those cells that we've engineered don't persist in the setting of these epithelial cancers, and we're trying to figure that out using exactly the approach that you alluded to.
Yep, over here.
How available is the CAR T cell therapy? So, let me see. Prior to Victoria's center being able to manufacture and produce these CAR T cells, CD19 CAR T cells, the only option was for Canadians to go to the US to get a commercial product. And at the time, there are now 5 products that are commercially available, but at the time when we started, there were only 2 available. And the sticker price, if you went down to the US to get this, so the manufactured list price is $500,000. And that was the upfront cost. You basically put a down payment down. And so, but these are life-saving treatments. So the provinces were kind of making decisions. I don't know how those decisions were made about who to send to the US to get the therapy, but it was a small sliver of patients that could go down because of cost. And that was really a driving factor because, in our healthcare ecosystem, a $500,000 treatment is just astronomical. And so one of the driving factors for why we started this program and built this facility was because we wanted to make sure accessibility for Canadians was not limited by cost. And so based on our best calculations, we have been able to reduce the cost, by about 50 to 60%, something on that order. Yeah. Yeah. So right now, the CAR T cell that we have made in Canada is not yet Health Canada approved. We're going through those steps of getting that approval. And so the way that you can access it is through a clinical trial. So it's still in what we call the research or experimental therapy domain. And because we have now proved that it's safe, it's efficacious, it's as good, if not better, than the commercial products, then the next step is to apply for market authorization. Well, we have to deal with government, so I don't know. I mean, the process has started, and it's a long process to get new therapies approved. But yeah, we're hopeful that it could be approved so that it will be what we call standard of care, right? So patients who have that certain type of leukemia or lymphoma, that's what they will get as the standard, paid for by the province. That's the hope.
Yes.
When you're talking about these sorts of, I guess what I would call exotic treatments, what are the opportunities for getting price down by scaling?
Yeah, so what are the opportunities for price down by scaling? We have a whole team that's looking at the economics of this, right? How can we reduce the complexity of these exotic treatments in such a way that can be affordable and accessible? So you've heard the term personalized medicine. This is the penultimate of personalized medicine because you're using the cells of that patient and only that patient. So every time a patient comes in, we have to manufacture a product for that individual. That is the definition of a personalized medicine. So many of us in the field are trying to figure out how to make what we call an off-the-shelf CAR T cell. Right? So you generate a library, right? And you bank it. And then when the person comes in, you don't have to make it again. You grab it off the shelf and give it to the patient. And I think that will help. I don't think it'll solve the cost issue, but that will certainly help the cost, bring down the cost of therapies. But we're still quite a ways from being able to do that. Yeah, it's a great question.
Okay, last question over here.
This idea of banking, is it dependent on blood type to be able to create a library? Is there one blood type that lends itself more to the making?
So is there a blood type that lends more to being able to create this bank of cells that you can just pull off the shelf for a patient that comes in with this leukemia, with this certain type of leukemia? So, this is analogous to doing any kind of organ transplant, right? You need a match. So that's one way to do it, right? You create a bank of cells that have a match, to somebody. And then you have to, what we call, type the individual when they come in. So that's one possibility. Another possibility using this CRISPR technology is that you remove the genes that allow those engineered CAR T cells to be rejected by the host. Okay? So you snip it right out of the cell's DNA. So now the host does not recognize that engineered CAR T cells as foreign. And that's another approach you can use to create an off-the-shelf product.
Okay. Well, let's give Dr. Julian Lem a round of applause for an exciting talk.
You can keep the props.
Again, thank you very much for coming out, and happy holidays, and we'll see you in the new year for the next Café Scientifique.